Good afternoon. We are here for the fourth webinar of the Open Science Monitor. People are still entering the room, so I will just start briefly with more operational issues. We're very glad to have today a presentation of one of the most inspiring case studies that the Open Science Monitor has carried out, Open Targets. My name is David Osimo. I'm Director of the Research at the Lisbon Council, and I'm also the coordinator of the Open Science Monitor. Today, we're going to have 1 hour of a very nice presentation on what is Open Targets, and we also will have room for question and answers. So every one of you will be able to see on the display a question sign. Feel free to submit question at any time. I will gather the question and put them to the speaker. I think we can start now. Open Targets is the case that we're going to present today. As some of you know, the Open Science Monitor has presented a series of webinars, and we have looked at how open science is being used, the practice of open science in academia and in different research centers. Today, it's particularly interesting, and I'm particularly glad to have you all here to illustrate how industry deals with open science, benefits and invests in open scientific endeavors. And this is why today we have here Open Targets, an inspiring initiative by a consortium of industry and research centers. As I said, today we're going to have 1 hour of presentation. The agenda is as follows. We are going to have now a brief introduction by René von Schomberg, a team leader on open science at the European Commission, who is in charge of the Open Science Monitor. We then have the chance to listen directly from Dr. Ian Dunham, the director of the Open Targets Initiative. And after that, Laia Pujol, a partner of the study from ESADE, who has carried out the case study on open targets, will present her perspective, her analytical perspective on how open target fits within the overall open science phenomenon. And then we will have a question and answer session. As a start, for those of you who don't know, I would like just to briefly introduce the Open Science Monitor of the European Commission. The Open Science Monitor, I will share the link, don't worry, gathers a mix of qualitative and quantitative information about open science. So we have data and case studies on open access, open research data, and open collaboration. In every session, you will be able to see key statistics of uptake of open science and case studies, a set of case studies. And you can see Open Targets here. So you can already actually download Open Targets directly from the website. I will now give the floor to René from the European Commission in order to present the initiative of the Open Science Monitor in general and how it fits within the European policies. René, you can speak.
Okay, thanks David. It's really a pleasure to do this. I always say in Trumpian ways, this is the greatest open science monitor in the history of mankind. And so why we do this? Well, you know, as David already explains it, the monitor in quite a comprehensive way tries to capture the recent trends in open science along the 3 pillars of open data, open collaboration, and open access to publications. Now, what is less highlighted probably in our discussions on open science in the recent years is doing open science in practice, how it changes as we say, the modus operandi of research and innovation. For the new framework program Horizon Europe, we actually flagged open science as a factor which will make science more efficient, because we can share resources better. We will make open science more reliable, because we can verify information better and in a more thorough way. And finally, also make science more responsive to societal challenges by working in open collaboration and being sensitive to societal demands. So there is a rationale for open science there. You can say that open science may actually make science better, or maybe it's even the best way of doing science. And I think an highlighted fact from the open science dimension is actually doing open science in the form of not just open outputs, so open data as an output of research, but also using open inputs for research. So again, open data as input for research, and also open collaboration. And I think today we will have a very important case actually where open data plays a role. I think also in a clever way of combining different data sources in order to advance drug discovery. So I really look forward to know more about this case. It's certainly one of the things in which we can say that open science in operation will make science better and also lead to better outputs. So I think I will leave it by that and look forward to the presentation of Ian, as well as enjoying the conversation later on.
Thank you very much, Ian. I will now give you the floor and make you a presenter so you can display your presentation. I will remove my camera for the sake of bandwidth.
Great.
Ian? Yeah.
Hope you can hear me okay.
Perfect.
So many thanks for this opportunity to talk to you about open targets and the mission that we have to try to improve drug discovery. So I'm going to talk approximately about 20 minutes, but I'll talk about the problem that we want to solve, and then a little bit about how we form a consortium to work on that problem, and then some science about the approaches that we use, and then at the end a little bit about the importance of open data. So you probably will know already that drug discovery is a difficult process, and you have to start a lot of drug discovery development projects in order to get one successful drug at the end of the pipeline. And the problem with that is it costs you a lot of money, particularly in clinical trials, and that return on investment is gradually decreasing in the pharmaceutical industry. So what we want to do is to try and improve that process, to reduce the attrition in projects by picking more successful projects earlier on in the process. If you look at why drug discovery projects fail or clinical trials fail, and this is old data, but it's backed up by more recent data as well, you see that roughly half of the time in phase 2 and a little bit more in phase 3 clinical trials, drugs fail because they don't actually work better than placebo or the standard of care in the trial. In addition, they also fail significantly because of safety risks that are found in the trial. So between efficacy and safety, you get a very significant attrition in clinical trials. And because of the way that the drug discovery process works, if you're trying to make a drug against a specific target, the choice of the target happens very early on in the process. And when you've made that choice, it commits you to a number of processes that you need to go through in order to get a drug out of the end, including the clinical trial. So that choice of target is a key decision when initiating a drug discovery project. So that's the background as to why we're interested in this problem. And Open Targets is a consortium that's come together between two academic institutes, the European Bioinformatics Institute, which is part of EMBL, and the Sanger Institute, with 5 pharma partners, GSK initially, Takeda, Celgene, Biogen, and Sanofi, with the idea of trying to transform drug discovery through enabling better systematic identification and prioritization of the targets that go into the drug discovery process. So there's a little bit here on the makeup of the partnership. I've already told you the partners, but you can see in there that each of the partners brings expertise into the consortium. The academic labs have expertise in genetics, in high throughput genomics, in life science data, and bringing together large public domain resources. And then the pharma partners obviously have expertise in developing drugs, but overall a common interest in neurodegeneration, in oncology, and immunoinflammation. Open Targets is based near Cambridge in the UK on the Genome campus in Hinxton. There's a number of other things that happen on the campus that make it a very attractive place to be. Obviously, the two institutes, Sanger and European Bioinformatics Institute, but this is also where the Genomics England sequencing takes place. And we have about 10 companies in a biodata innovation center, just a few meters from where I'm sitting at the moment. Sanger Institute is obviously a world leader in genomics, in human genetics, in cancer, and also now in using the new gene editing techniques and single cell RNA sequencing techniques in cellular genetics. So this means that there's expertise in high throughput genomics and genetics techniques that we can use. European Bioinformatics Institute is really probably the world leader in using big data in biology and providing a trusted source for data in the life sciences. There's some research activity that takes place on campus and about 650 staff from 60 odd nations here. So our vision as Open Targets is to, as I said, is to try and improve the success rate of drug discovery programs and be a world leading center for identifying and prioritizing targets. In order to do that, we need to understand the processes that have a demonstrable effect on disease, and we need to use experimental and computational approaches that are at the cutting edge to address these problems. The types of technologies that we use are large-scale cell line connections, both in cancer and in normal cells in terms of induced pluripotent stem cells. We use gene editing, we use single-cell genomics, and we use complex tissue culture, including organoids. On the bioinformatics side, we are very much interested in genome-wide association studies in genetics and high throughput, high performance computing in order to address the issues of integrating large-scale data. So when I talk about identification and prioritization, what do we mean by that? So this slide illustrates the approach in a very simple way. The first step is identification, where there's about 20,000 protein coding genes in the genome. And what we want to know is which of those genes has an association with the disease of interest, and can we rank those in terms of the degree of association? So can we tell which has causal association through experimentation or integration of data with the disease? Types of approaches we use there are large-scale data integration, genome-wide association studies and genetics, and also synthetic genetics in cell models, where we introduce mutations to observe the effect. And then the second step is prioritization. So from that list of targets that are associated with disease, we want to know which are the most likely to be able to make a drug, able to make a safe drug, and will have the right physiology to be used in a drug discovery program. So this is the area where we bring in additional information to try to prioritize the choices based both on their efficacy and other factors. So Open Targets as a whole tries to work as a collaborative consortium, and we try to build projects together that involve all of our partners. So we don't just take money from the pharma partners, go away for 3 years and then come back and say what we did. What we do is try to have active engagement between the scientists in both the academic partners and the pharma partners on projects. So when we build the projects that we work on, we collect ideas from each of the partners, from the scientists involved, and try to workshop those ideas with each other. We bring the scientists together, often in the same room, but by teleconference as well, to try to discuss what's the burning issue in the area that we're interested in, what are the approaches that will be used at cutting edge, and what can we do as part of this consortium that cannot be uniquely done elsewhere. And then we identify projects that we want to do, and we commit to those. And we have about 50 projects running at any one time. And we have a more formal process that goes through that, but that is essentially how we pick the projects that we're interested in. So we think about this problem of target identification and prioritization as a kind of circle where public data can be used by data integration to form therapeutic hypotheses that might be used in a drug discovery program. In addition, we want to generate new data in our key therapeutic areas of oncology, immunoinflammation, and neurodegeneration, and release that back into the public data to enrich the data ecosystem. I'm going to just talk a little bit about an example in this experimental area now that's been published recently. So in this project, we used in the lab of Matthew Garnett in the Sanger Institute 324 cancer cell lines, and we subjected them to gene editing in a method that's called whole genome CRISPR dropout screen. So what this does is we edit each of the genes in a pool of cancer cells, and we look for the cells which die in those populations, so are no longer able to grow, and identify the guides that are lost with the cells that die. And those guides are the CRISPR guides represent genes that are essential for the growth of the cell line. That could be two things. It could be because the guide is, or the gene is constitutively essential, or it could be that it's only essential in the context of the cancer cell line. And in fact, the latter ones are the ones we're interested in. So if we can identify these genes that are context specific, we can then try and prioritize them by putting additional information that's relevant to drug discovery alongside that data. So here in this schematic, what we're showing is we identify the genes that are essential for fitness in a particular cancer. We then integrate them with expression data and pathway information, and with information that tells us whether it's likely we can make a drug. Either there is a drug already, or it's likely that we can make a drug to this particular gene. And that prioritizes the targets that we might be interested in. So in this case, in colorectal cancer, you can see that KRAS and novelly the Werner helicase gene have been identified as both high priority and currently lacking drugs. And in fact, in this study, they followed up on the Werner helicase gene to identify it as a target that's likely to be of use in microsatellite unstable colorectal cancer. And this attracted a lot of interest. It was released in the spring this year, so a lot of press coverage on this paper. All the data is released through both a project website and through the tools that we have at Open Targets. So this is generically the way that we try to use experimental approaches to identify targets. We have a range of different cell types in different types of culture models, either organoids or mixed cultures. We use gene editing extensively, and then we try to identify genes that are selectively detrimental to particular phenotypes. So, for instance, cell death or something that we can measure with a fluorescence assay. And out of that experiment comes a list of targets that are relevant to the phenotype. Okay, so now I'm going to move on to discuss a bit more about the data integration and the open data side of Open Targets. So one of the things that we identified early on is that there was no place that a drug discovery scientist could go and identify all the evidence that was relevant to whether the target they were interested in was involved in disease. And we realized that we could bring together data across a number of public databases that were based at the European Bioinformatics Institute and elsewhere, and integrate, though, the evidence that comes from those databases around this target disease model. And what we did was to build a system that essentially looks at each of the data and our databases and asks the question, what's the evidence that comes from those data? And brings that into our platform. We have to do some mapping to make that work. We have to have a common language for disease in terms of an ontology, and we have to have a common set of identifiers for genes and proteins. And we also have to have a structure for identifying and scoring the evidence that we bring together. We have that backbone of data that we then provide through an interface which we call the Open Targets platform at targetvalidation.org. You can see some publications on that. The database is live and you can use that URL. It's publicly available, free to use. And what you can do there is you can search for either a target or a disease and find out the diseases or the targets that are associated with the query. So for instance, if you ask the question, which targets are associated with breast cancer, you get back a heat map with the density of the blue color indicating the strength of evidence for a particular target to be involved in breast cancer. And it's shown in this little heat map at the bottom here. So you can see that some of the usual candidates, BRCA1 and 2 and so on, at the top of this list. You can also do various kind of prioritization activities. You can filter the evidence by the type of evidence that's available, by which pathway or target class it's involved in, or whether the gene is expressed in a particular tissue. So that data is all available, and it's a very large data integration process behind the scenes that brings it into the platform. We also provide tools within the platform that enable drug discovery scientists to ask the questions that are specifically relevant to them. For instance, whether that target is likely to be tractable by a small molecule or an antibody approach. The other side to this is that we also note in the literature is that drugs with a strong genetic link between a target and their target and disease are more likely to succeed in the clinical trials. So this is data from a paper by Nelson et al., which shows that when the target has a genetic linkage to the disease, either through rare disease genetics or through GWAS, it's up to at least twice as likely to proceed through clinical trials. So in order to meet this demand, we set up a database that essentially takes all of the world's GWAS data as summary statistics and integrates it with other data on expression and gene regulation to identify the genes that are likely to underlie the genetic signal. And we put that into a database that's called Open Targets Genetics. And this will feed genetics information directly into the Open Targets platform. So here is the front page of this. It looks similar to the platform. It's available at genetics.opentargets.org. And you can search here for, again, a gene or a trait, but also the variants that link the genes and traits together. And the way this works then is that the genetics platform aggregates evidence for targets, which goes into the Open Targets platform and improves the scoring there. The Open Targets platform also provides information into the genetics portal to investigate the details of the evidence that's involved. So those are two products that we make available freely, but also to our partners. You might then ask if Open Targets is a pre-competitive initiative in which releases open data, what's the benefit for the industrial partners in actually being involved in the partnership? Well, firstly, they get to direct the research direction or have input on the direction. They have input on the therapeutic area, focus and strategy. They have access to all the data, methods and technologies to support their internal research programs, which can enhance the internal R&D capabilities of the companies, de-risking emerging areas. They work with world-leading teams in genetics, bioinformatics and cell modeling, and also with their equivalents in the partner companies. And so there's an exchange of information. And that really gives a community that can work on this process of target selection. We have quite a lean structure with a low bureaucracy. So most of the money that's involved gets spent on research projects. And we also have a clear IP process for determining whether there is any IP that should be protected. And there's also, of course, reputational benefits for the partners through being involved in the partnership and also in open innovation. I just probably should say something. What does it mean about to be when we say open targets? Well, all the data that we generate goes out into the public domain at the very latest when it's published. We also release the data integration tools as open source. They're freely available. There's no account logins. So anybody can use these. They can set up their own version of the tools as well. And we welcome collaboration on those tools. So just thinking a little bit about policy implications of this kind of structure. So one of the things about these databases is we can only do this if the data is freely available and accessible to us. Because what we're trying to do is we're trying to summarize essentially the world's knowledge on the evidence between a target and a disease. And what we want to be able to is summarize that knowledge across all data sources. And so that sustainable integration is only possible if the data that's being generated across the world is available and sustainable in public databases. And if you think about that more generally, again, if we want to make these open access, open data resources, we have to have the data from projects that are funded across the world. So for instance, from the European Commission, things like IMI or the Horizon projects, to become available publicly so that we can integrate it, so people like us can integrate that data into sustainable public knowledge. Okay, so I'm going to finish up there just to show you a picture of a recent meeting with many of the team who work on our projects represented. Thank you.
Thank you very much. This was really interesting and really intense. I'm sure everyone was looking very closely at the screen, just as René, that we saw, whom we saw, for understanding. We are starting to receive some question. Maybe I can, before giving the floor to Laia, I can share with you one question that we received, and I will share it as it is because I'm not sure I understand it fully, but I'm sure for you it will mean much more. So the question is, how do you search for evidence? And in particular, is the coverage in this database search for the number of times it is mentioned found in the database as one possible criteria? Do you understand this, or we can ask for clarification?
Well, I can answer that in several ways. So what we do is we have in our mind a model of what kind of evidence is relevant to the specific question that we're asking, which is whether a target is involved in disease. And so we identify databases that have that type of information. There are different sorts of evidence. So there are some places where there's curators that curate from the literature that a target is involved in disease. There are other places where, for instance, the database ClinVar is a database of just reported mutations that are found in individuals with a rare disease. And there we have different types of, we have just single instances of an observation. We also have GWAS, which is a probabilistic association. And we have text mining, where we might mine the literature and look for occurrences of sentences that relate a target to disease. So each of those methods has a different scoring scheme where we try to internally to the method to be consistent. So we have to be able to rank the data internally consistently within each data type. We then bring it all together and we have a view on what the relative importance is of the different data. And that view, we actually want to try and gradually evolve that by using approaches like machine learning to try to establish what the weights should be. So I think that kind of answers the question, but so when we don't just use a raw count of how many times something occurs, we have a model in the background that models the data and gives it a score that's internally consistent.
Yeah, what is clear is that this is not just publishing and reusing data. What clearly emerges for your presentation is the amount of work it goes into enriching and analyzing and make this data meaningful, and not only meaningful and relevant, but also accessible. I was struck by the simplicity of the interfaces that you have. I think rather than going to the next questions, I would rather move to Laia, who has studied and published the Open Targets case studies for the Open Science Monitor, and she can bring in her perspective on how this case fits within the overall movement of open science. So I will now take the floor from Ian. Okay, just a second. Here it is. Laia, the floor is yours.
Okay. So thank you very much. I mean, I wanted to share a little bit. So why we pick open targets, and we thought that open targets would be an interesting case for the Open Science Monitor. So as David and René said before, as part of the task that we need to do, we need to identify and monitor the different trends under open science. Open science is an umbrella concept. So It integrates open access to publication. It refers also to open scientific data, so how scientists and researchers share and reuse public scientific data, but then also is about open collaboration, so sharing open scientific code, even open scientific hardware, instrumental tools, open review, citizen science, and different trends. So when we were trying to put together the different evidence about what's going on in open science in Europe, we found that we didn't have enough metrics about open data, which is very relevant. So the approach through selecting different inspiring cases that allow us to better understand how data was publicly shared and then reused by different organizations was key for us. So this is how open targets appeared as a relevant case, I mean, that could illuminate us. So as Ian Dunham shared already, so why was it important? Because open targets gave us the opportunity to look at this kind of virtuous cycle that you have created through data generation, so how you generate scientific data through different experimental projects that open targets run in collaboration with different companies and academic partners. But then we see how it's integrated then into a platform that third parties and other organizations, SMEs, and plenty of other and the scientific community is reusing and benefiting from it. So we wanted to better understand this whole process, and also, I mean, the first place, how would companies agree to generate data and share relevant knowledge that was going to be publicly disclosed after a while. So I'm going to share, just because of matter of time, and I think that the questions are, and the discussion could be more interesting. So some of the takeaways that we took, I mean, from the case. So basically, first of all, we wanted to better understand what is shared, I mean, through the different parts in the consortium. So why would companies agree to share data and information, and what is exactly what they are sharing so far between them? So we try to structure the drug discovery process to better understand what is defined as pre-competitive. So something that, what is this kind of space where companies feel comfortable sharing and different knowledge and data, and which are the different, and when this pre-competitive stage turns and goes further a competitive process. So as it was shared in the previous presentation, is about, in this case of open targets, it was about putting the boundaries of what is considered pre-competitive, that is about the target identification. So identifying a target that is safe, that works, and have different characteristics. So this is the space where companies feel comfortable, and then what they're going to do afterwards in their pipelines through the drug discovery process, of course, this is going to remain close to their doors, because it's where they're going to be competing with each other. So how they're going to fight this target, I mean, to overcome the disease. Then an important thing was, okay, and then how? So besides what is shared, so how they work together. And it was very interesting, this kind of, I would say, alternative approach of how they put together different resources and pull out and exchange ideas. So as Ian Dunham said, it was very interesting. It's very interesting approach to see how the different academic partners and industry partners sit together, brainstorm, share some ideas, and how these ideas then are combined through a brokerage process, is how we call it. I mean, thanks to the Open Targets team, so that they help to cluster and put together the different ideas, looking at the commonalities, and then how these ideas convert to proposals and then to projects that can successfully advance innovation and what we know about targets. Another relevant idea, this is from a governance perspective. In order to make all the different organizations comfortable, industry partners, etc., you have to come up with a modular architecture, in the sense of what are we going to share, what is going to be remained protected. So it's very interesting to see. So the platform is public, everybody can reuse the data, and then there's part of the data that is shared, I mean, through the experimental projects with partners from minute one. And then we have also the work that the different organizations, but not only the partners from Open Target, but also like different stakeholders are going to do privately. So they're going to take the platform, the information, the evidence that is out there, and they're going to work in advancing knowledge that is going to be proprietary, and they're gonna like exploit commercially later on. So this kind of articulation of public and then what is shared across partners, and then it's very interesting because it solves the different interests and makes possible this kind of convergence. So let me share some final takeaways, I mean, before the discussion. So the case illuminates us in the sense that we moved away from this ideological debate about being pro-open or pro-closed, but this case shows us how you can be smart in being open. So there is, and we know this from the literature of business and management, but also there's an inherent tension within the goals of the scientific openness. So academics, we want to share as much as we can, and then the commercial exploitation, which is necessary, I mean, for companies to exploit later the knowledge that is produced. But it's not then about being pro-open or pro-closed, but it's finding sophisticated solutions that can balance the different interests. Then the second idea is also it's important to define this common ground. So companies are reluctant to share knowledge and data that move towards the heart of what makes those companies competitive. But you can accommodate and you can make them agree to share if you can define these these boundaries that are far from the market exploitation. So in the case of drug discovery, it's about targets. So you need to define what is like far away and where companies like could feel comfortable, I mean, disclosing and sharing. And finally, and this is one idea that is stressed and very important in the case, and then also David picked it up very nicely. So what Open Targets show is that data publication and sharing public data and releasing, uploading is just the beginning. So if we seek to reuse scientific data by scientists, but also by other companies, by SMEs, different players in the ecosystem, there are different conditions that need to be put in place. So we have in Open Targets a very user-driven platform design that was done. So there's a paper also and is available in the Open Targets website about how you build actually this platform very friendly, because it looks like Google search very simply, and it follows the different workflows that then users implement, I mean, when they are looking for evidence. But not only is about the design of the platform, so this is also the complementary support that the platform teams gives to people who are trying to reuse the platform. So there are webinars, there are also kind of support on how to make possible to reuse the public data that is out there. And not only this, but third and finally, I'm going to finish with this idea, still, even if all these conditions are met, still industry partners are willing to engage in a close interaction with academic partners to generate themselves data, because this gives them insights and kind of like advantage, I mean, to reuse and to work and to play with such data. So with this idea, yeah, I'm give the floor back to David.
Thank you very much. We have had a very rich set of insight. I would like to give people the chance to pose some questions. We already have received quite a bit. For all of you who are watching us, this is the time where you can submit questions. In the meantime, just let me point out a few housekeeping issues, and mainly that there are ways that we can keep this conversation going. We have a group where we discuss indicators and case studies on LinkedIn. It's called the Open Science Monitor group. You can find it there, and obviously the link to the Open Science Monitor itself and my personal email address for any information. So thanks a lot. Now we have received some questions that I would like to share, and it's a question to Ian. Looking at all that Open Targets has delivered so far, what do you consider now the major impact, the single thing that where Open Targets really made a difference?
Okay, that's quite a complicated question because we work over several areas, but if we are to judge where we are successful, what we want to know is, are we making a difference to the choices that pharma partners and other pharma companies make when they choose targets? And that's actually quite a difficult one to measure because you have to, that information is proprietary to some extent. So we can look at proxies to that. So we can ask our partners if we made a difference to things, and we have some case studies where that is the case, where we know they've chosen targets or sometimes rejected targets on the basis of data that we provided. We also can look at the uptake of the Open Targets platform by other companies, and we, again, because they talk to us about the platform, we know that it's being used in a number of other pharma companies that are not directly our partners, so we're making a difference there as well. So I think, you know, that's where we want to make the difference, and I think we are. In some senses, it's hard to measure though because of the reasons that I said.
One additional question is about the issue of trust. I mean, obviously, having companies to collaborate, it's difficult. And in general, this is a problem that we see for open science everywhere, but in particular when it comes to areas which are closer to the market, when the IP is very strong. And the question is really about how build the trust. And a very specific detail in your presentation is, did location matter? Does location matter? Does physical location matter? The fact that you have this kind of cluster of companies and industry together. You know, the rhetoric of open science is all about global collaboration, how everyone can collaborate with everything. But maybe because open science requires high level of trust when it comes to sharing data, maybe co-location plays a role in this.
Yeah, so I think there are a couple of aspects to that. So the first one about trust is within the Open Targets consortium, we actually have a confidentiality agreement that covers all of the partners. And what that allows is the scientists from the pharma companies and from the academic labs know that they can freely talk with other members of the consortium under that confidentiality agreement. So that gives a level of trust when they're working on specific projects. It just establishes the baseline. I think the idea of proximity is also important. At least you know, several of our partners are actually have major research centers in the US, so we're not completely proximal to them. But what we do in order to develop that trust is that it has to come from interpersonal interactions, and so we enable that by bringing scientists together. So myself and other members of the executive team travel to the other sites in the US or Japan to talk with scientists on the partner sites. We also bring scientists from our pharma partners onto our campus regularly. So 3 times a year we have a big meeting where they come together and they can make those personal interactions that then allow them later on to contact via email, and the trust is established. So I think that is an important part in this particular model in making trust happen.
Great. You mentioned the need for open data to work and to generate new data. But what would you say to the European Commission, and we have René here, for the Future Horizon Europe, and how do you see the influence that the decisions taken in the future policies? We now have a new commission in place, or soon we will have a new commission in place. What would you tell them?
Well, I think that one of the frustrations that we have had working in this area, for instance, in the area of drug safety, is we know there have been a number of projects that have looked at that through instruments like the IMI and have generated data, but that data is private and proprietary. Now, we feel that if we could get that data out into the public domain, we could actually have an impact that would enable everybody to be able to make better choices on safety, for instance. So I think that's an important thing, is that the outputs of these European projects need to eventually be made into sustainable databases. And that means that you have to budget for that sustainability in the long term, and you have to provide incentives for the scientists to make that happen, other than publication, because publication, although it's the de facto standard for releasing data, as you said, it's only the start of that process. If you want to reuse the data, it needs to be accessible in some way.
That is great. We are now approaching the end of the webinar. I don't know if anyone has a last question, even on the panelist, or René. I don't see you, but I trust that you're still there.
Yes, I'm still there. Actually, I would like to say something as a response. Let me see if I can try to get with this webcam on again. Yes. So no thanks again for facilitating this webinar. I find it really interesting. Just a very short response to Ian's point on having data more open. Actually, the Commission wants to go further than just having sustainable data available. So what we actually try to do, of course, with the European Open Science Cloud, which formally was launched last year, is actually to have data available, also mandatory, of course, for the next framework program. Data should be, everybody who benefits from publicly funded research should a priori deliver its data. But the cloud will actually facilitate the possibility of data integration across disciplines and also across nations. Of course, the whole sustainability issue lies under the cloud issue, so it's of course an enormous investment for member states and the Commission to do that. But we have committed to that actually already since 2016. So I think there we are on track. I think actually what happened here with this example is actually something what in the future when we have a cloud, because if you have data in the cloud, of course, it's something else still to use this data and provide services. So I see an initiative like from Ian's Open Target as a service which you can actually add to the cloud, and where you can do also the data integration, what you actually also now do here in your context already. But of course, if we have a European Open Science Cloud, you can do it even on larger scale.
Great.
Now, I think this. So this is. I mean, there the open science has, of course, a gradually growing dimension. But the question back to Ian actually is that my understanding of clinical trials actually is that in phase 2, 90% of the trials fail, and this is for one reason. It is because they have, you know, the quality of the data is very bad. So I was actually wondering whether in the context of your activities, you found actually by doing this data integration also that you improve existing data, or that you come actually to a track where you have to launch new type of research, because a lot of the data out there is actually, let's put it this way, crap, which is not good, which you cannot use.
Yeah, that's quite a general statement. So one thing I should mention first of all, that all our software actually is packaged in containers, so it all goes on not the European cloud, but it's installable in Google Cloud or Amazon Web Services, and would be transportable over to the European Open Science Cloud as well. So that might be a good use case for it. The question about replicability, I think, is an interesting one. We haven't studied that specifically. I think it's also true that the data from clinical trials is actually quite difficult to get hold of. We get the outputs of that data, so we know the result of the trial, but it's actually quite difficult for us to parse that data. What we haven't been able to do so far, so we've tried a number of kind of models to predict trial outcome in retrospect, and it's actually quite a difficult problem because the data is very sparse, both the outcomes and the input data. So we know that there are some pieces of evidence that give you more, you make it more likely that the trial is likely to be successful. So genetics is one. There are some others as well, but for any individual trial, it's actually quite hard to predict.
Excellent. Thank you very much for all your contributions. I think this was both an excellent case of open science and a very important one in terms of where it is applied. And as a social scientist, we always suffer from the envy of life science. So we would love to have an open targets for policy analysis, but that's too early. So thank you very much, and look forward to continuing this conversation online. And thank you in particular to Open Targets and Ian Dunham for the presentation. Goodbye, everyone.
Goodbye. Thank you.
Bye-bye.